See, this is what happens when you kick me out. You have to hear me again.
Yeah, so Prashant, you were saying?
No, I was saying this is what happened when you kick me out.
No,
You have
just
to hear me again.
I was just inviting you to continue where you left off. Sorry for that. Sridhar gave us more time to chat. I didn't know that this was Resolve's pitch, to be honest.
Yeah, that's what, like, when I talked to them, and that was kind of very attractive to me. Right now we are just running Visium. We don't, another facility has a phenocycler. Now we are kind of, in a situation where we are kind of debating whether it's going to be, Cosmax, Xenium, or Resolve. And I think Resolve seemed very interesting to me because of this feature because they said they have probes for entire transcriptome for human and mouse, and you can pick and choose whichever set of genes you want. And I don't know, I don't remember exactly what was their minimum number of slides you have to run. It was like not a big number, it's a very small number. So if anybody has any more information on that, anybody using that in this group?
Prashant, I don't recall their kit sizes, but we did generate a data set with them about a year ago, and the data at least looked really good.
Yeah.
We decided to not go forward with them because the instrument was expensive. I think it was like $150K.
Yeah, and then even now they're more pitching as like a service lab. They're not selling their instrument aggressively. So yes, that's one of the things which kind of holding us back to proceed with that.
I mean, I don't know about other people's experience, especially those who are doing Murph-Hope, but we had a Murphish. I mean, we have still Murphish, which we actually in-house developed system, and we are struggling with that for last 2 years. So basically, getting a good data consistently out of that system. And what I heard from some other people, and maybe I may be wrong, that even the VisGene, which is commercial platform, has often it breaks down and there are issues with the system itself. So I think the Morphish as a technology has some kind of an issue which needs to be resolved at some point.
So instrument-wise, we have the first and the second Merscope that left the factory because the first one didn't work. But since then, I think our downtime was not very long, to be honest. But it doesn't make, even though we have more than, we got it, I think, July 2021. So it's quite a while for this technology. The assay has not become much easier for us. So every tissue, even if it's a tissue, unless it's brain, every other tissue we have to optimize. And usually with the clients is very difficult to explain to them that we will burn $1,000 or $2,000, $3,000, $4,000 runs, $5,000 runs, just to get it right. And with FFPE samples, FFPE is a very generic thing that means very different things to different people. How long, what's the process, how long it stayed, how has it been collected, even who have experience working really well with one tissue that is FFPE, even if we get the same tissue from a different location, we might need to optimize again. So I don't know if other people's experience has been like that, but it needs a lot of hand skill to get it working well. And time and money. So for the second round, I wanted to ask, just because I'm very curious, your number one challenge and number one goal for 2024. We have discussions in the lab now because October 1st is our fiscal year beginning, and we're ranking our challenges and ranking our goals for the year. I wanted to ask if anybody would like to share what's your number one challenge for now, and what's your number one goal. For us, number one challenge is automation. So we need to make this assays high throughput or as low involvement as possible. And that's a goal and a challenge at the same time, but that's where we're investing 2024. Would you like to share your goal and your challenge for the next year?
Yes. Well, I mean, the Jessica Podner, I'm at the University of Texas in Austin. So I kind of the goal here at UT is to bring in an in situ platform to campus. Of course, right now, with multiple things to choose from, it's just like, getting the right information on like, what do we choose? And, I kind of appreciated what Anoja said, because we also do a lot of non-human. And so it's important for us that it's something that's compatible with lots of things. But I kind of wondered, like, in order for us to get this goal, like, I appreciated how she said, like, okay, non-human and 10X was familiar. So like, kind of like, if anyone has any input on like, how have you decided what platform's going to work best? Like, are you listening mostly to your faculty members and the researchers? Are you trying to like listen to like one specific group that might do the most projects? Like, how is anyone making these decisions and to feel comfortable with that purchase?
That's a
I mean,
great question. Yeah, who would like to take it?
One of the things that I thought was, especially for non-human, non-mouse organisms, probably the Curio kind of a platform is a better one, because it don't have subcellular localization and those kind of information. But when it comes to cost, and especially the custom probe, developing a custom probe panels and those kind of things, I think it's always good to look for something whole transcriptome based platform. So that is where I thought Curio with 10 micron resolution is a pretty good platform to start with, especially for non-human, non-mouse groups. For actually, the biggest challenge was slightly different. I'm from medical school and most of our clients are from medical school in Yale, and they are using tissues like kidney, retina, aorta, and heart. And these are like tissues that are pretty difficult to work with, even for sectioning and as well as for the in situ. I mean, any method. It's not about one method or two. So that's where we are actually looking at, like, if it is brain, everything works. Then can we actually make these platforms work beyond brain and work consistently beyond brain?
Yeah, we'd love to repackage the discussion into two questions. One is, when you have decided I want to go in situ, how do you decide between 3 or 4 tomorrow or 5 different offerings but they might be similar as instruments, but very different products and might have different scopes and applications? And the second question is, now that you have Curio, Vism HD is right, up the alley, and you have complete genomics and more other NGS-based assays coming in at high resolution, how is that informing your investment in a platform? So you have two things. When you have decided for in situ, how do you choose one? But when you know that high resolution NGS-based assays are coming, how do you invest in kits or boxes? Emily?
Just to speak to Jessica's question, when we were looking at the different in situ sequencing platforms, we're just comparing the metrics between them, and they're all very similar. But just from past experiences of purchasing platforms that weren't necessarily fully supported and like, I don't know, almost half-baked sometimes. We kind of go with something that is going to be reliable. That's why we kind of ended up with the 10X Xenium, just because we've worked with them previously, and most of their assays have come out fully formed, and their support has always been extremely helpful when they have fallen short on some things. So I can't speak to like NanoString or anything like that, but some other spatial platforms that I've used, we've had issues and then have gone to support, and they have not been very helpful. And I think some of these companies that are maybe more new on the spatial scene aren't as reliable as some who have proven themselves, basically.
Thanks.
Thank you. Lara?
Hey, thanks for, letting me speak. So I might actually be a minority here, but I really wanted to join this group. I am the managing director of the bioinformatics core at the University of Alabama at Birmingham. And obviously, bioinformatics complements all the wet lab biology. And I think to speak to your question specifically in terms of goals, and this is actually picking up on our goal for this year. We've been doing a lot of single cell analysis, all these other pipeline developments for a long time, but of course, spatial is the next big thing. So we're trying to do those same tasks for the spatial omics. but we have to do across the board as much as we can, which includes Visium, Xenium, and at the bare minimum, geomics. We might consider cosmics. We're not quite sure yet. The university may be, acquiring one. But I think that one challenge that comes to supporting all of these omics approaches really comes down to the bandwidth. How much can a bioinformatics core really provide for a university, the analysis solutions for all of these methods? I think that one point that I wish was as easy as it seems was that for the cores at UAB or in other universities that are acquiring these instruments to always contact the bioinformatics cores at the very beginning, because one challenge that I have faced and we continue to face is to really get data that is representative of the experimental designs the labs are going to actually have. Because if you go to, like, for example, the 10X website and you look at Visium data or Xenium data, sometimes they don't even have replicates. It's a single one, and that's not the kind of design that biologists labs are going to come to us. And that changes very drastically on the data analysis side, right? How to integrate properly, how do we take care of QC issues, etc. But the course may actually have that data, and we actually sometimes have to very proactively contact different folks to even get that data. And I come from a molecular biology background. I'm very familiar with what it takes on that end, and I like to actually be familiar with it. So that's a challenge for us, is the bandwidth aspect for across numerous spatial technologies, while still also providing solutions for bulk RNA-seq, single cell, and others, but also how to actually be able to improve that communication with other communities as they are bringing in this technology, so that we can have that data to start building our workflows from the very beginning and not when the labs have that data, if that makes sense.
Yeah, thank you. I tried to keep notes, and as a group, we're doing also the in silico work. So we know exactly what instruments and assays are coming up, and we prepare beforehand. But I can imagine also instruments just showing up out of nowhere, and you have to, like, suddenly invent people that have the ability to tackle the data, manage the data, move the data around. So I think bioinformatics, as I hear you, should be involved even before an instrument becomes a thought, not when the assay is being done. So and I think dry lab will be the major challenge after we iron out our production and the production ramps up. Then it's all dry lab. Anoja?
Yeah, so to answer Jessica's question earlier, how did we decide whose input we take in? One thing I did last year was invite every single vendor in this area to come and give a talk at my institute. And then following that, I sent an email. I mean, I'm not at a big institute. We only have 500 people. So I sent an email out to the institute and said, if you're interested in doing spatial, let me know. And then with that, I generated this group of individuals that are really in this space or interested in this space. And it consists of people from the different cores, like histology, sequencing, imaging, bioinformatics, and a bunch of PIs and postdocs. So anytime I need to get some, if I'm looking at a technology, like Cecinium, when I found out about their specs, I wrote to this group and said, hey, this is out there now. Are you interested in doing anything with this? And then, when I have interest, I would take that to the upper management and say, okay, we want to try this, but before we bring it in, can we generate some data? And can we throw some money at it? Because PIs obviously don't want to spend money on something that we're going to test, right? So those kind of discussions have helped. For my immediate goal, I would say it's to decide which instrument to bring in. And then future goal, like Lara said, I think bioinformatics is going to be the bottleneck. Even with Curio, we are seeing that right now. So for their 1 centimeter tile, I don't think it's available yet commercially, but they want us to work with DNA Nexus. And it's a lot of back and forth, and our bioinformatics team is super spread thin.
Thank you, Anoja. And I think on that note, we have 10 seconds. If there was a single advice we could give, is connect to the WhatsApp so we can continue our discussion. There we discuss projects, we discuss instruments, we discuss costs. So yes, Anoja's forum is very helpful. Great. having everybody here. Let's do this again, hopefully very, very soon.